Thank you for your interest in our whale photogrammetry measurement program. The programs that you'll need in order to be able to execute this analysis are VLC Media Player, MATLAB, R, and you'll need our whale photogrammetry measurement program, which you can download via our lab's website. If you go to the Geospatial Ecology of Marine Megafauna Laboratory website through Oregon State University, you can find the code under Research Projects and then Application of Unmanned Aerial Systems. And if you scroll down, you'll find the photogrammetry analysis code, which can be downloaded, and we do our best to keep this up to date with the latest version. So the first thing you want to do is open your video file in VLC Media Player, and most likely you'll have both a video file as well as what's called an SRT file, and that contains the metadata that is going to be important in order to be able to make these measurements. So I'm going to go ahead and open this file with VLC. And now, in order to get that subtitle track on there with the metadata that we're interested in, you can go to Subtitle and you can add Subtitle Track. This will let you navigate to where the location of that SRT file, and this will let you add the metadata to the imagery that we're extracting data from here. So we're first going to go ahead and extract a still of a calibration object. In our case, this is a board that we know is about 2 meters in length. And when we extract a still, we want the reference image that or the reference object that we're most interested in to be at the center of the frame. And then you're going to go ahead and go to video, and you're going to take a snapshot. Now that's going to send it to a folder which you can select. You can set the directory for that. And you can dictate where that snapshot is going to end up by going to Tools, Preferences, and then select Video. And under Directory, you can browse to the file that you would like that still image to get sent to. So we're first interested in extracting a series of images of this calibration object at multiple altitudes. You can tell the altitude from this barometer reading here. Subsequently, we also are interested in extracting several images of the whales that we would like to measure. So for the sake of this tutorial, I'm not going to review this video in its entirety. Instead, I'm going to jump to a section where I know there's a whale in it, but the analyst would theoretically review this footage in its entirety. So here we have a blue whale, and we would like this whale to be in the center of the frame, as centered as possible and as flat as possible with minimal curvature in the whale. And we would like to get it kind of just as or just before it breaks the surface. And again, here we would go ahead and take a snapshot. Now, once you have those snapshots, you can go ahead and exit out of VLC Media Player, and we can go to that, that whale measurement program file, and there's a lot of different program files in here, but what we are interested in is these MATLAB files that are whale calibration object measurement and whale measurement. So you can go ahead and open those up in MATLAB, and I'll start with the whale calibration object measurement. So if you open up this script, you can do that by navigating to the file and opening it. Then you can click Run. And it should give you a series of prompts. What camera was mounted on the drone that the image was taken with, And here I'm going to go ahead and click no calibration because we're going to deal with the calibration later on in this analysis. So I'm going to click no calibration. Next, you'll be asked to enter the camera focal length in millimeters, and this shows up as the default for Phantom 3 and 4, which are the drones which is the drone that was used for this image, but you can customize it as well. Next, you'll be asked to enter the camera sensor pixel pitch in millimeters. For reference, you can enter what your calibration object is, for example, a swim step on the back of a research vessel, or in our case, a board. Now we're going to enter the length of this calibration object. In our case, it's 2 meters, so 2,000 millimeters. And next it's going to direct us to where we had those stills, and we want an image at high, medium, and low altitudes with the scaling object in them. So now we're going to go to that, whatever directory you had those still images sent to. And here you need to select all files so that the PNG files will show up. And then you simply select the image and open it up. Next, you will be prompted to enter the sighting information. So this is from your survey data. For example, this might be sighting 1 of the survey. And then what flight it is. And this information is going to be very important for linking the calibration information with the information associated with the whale that you're interested in measuring. So for this example, we're going to say that this was flight 1 of the day. Next, you will enter the date. And once again, this is important for linking the calibration data with the whale measurement data. Now you're going to be asked to bound the scaling object with a square, right-click it, and then select crop the image. This is to give you a better ability to measure the image. So then we're going to click crop image. Now it's going to show us a zoomed in version. And you're going to be asked to click a point on each end of the scaling object. And then you're simply going to be prompted to press enter twice to continue. Now you're going to need to enter the difference in the takeoff height, or the difference between the water height and the takeoff height of the drone. And finally, you're going to be asked to enter the altitude information from the bottom of that still that you took. So if we go back and look at the image, we can see that the barometer is reading 21.8 meters. So that is what we're going to be measuring here. So now we've completed the calibration object measurement, and what should happen is in that folder where your stills were that you measured, You will now have a measured image, and you'll have this CSV file which has the measurement information in it. So this is what the output is from the MATLAB script that we just worked our way through. So now we're going to switch and measure the whale. So for this, you need to be in this other script, whale measurements. This is again in that same folder that you download with the program. And so just like before, we're going to click run and follow a series of prompts that are pretty similar to the calibration object. Again, we used a phantom 4 and no calibration. Again, we'll be prompted to enter the focal length, the sensor pixel pitch, and then we're going to select the images to be processed. So here we'll need to, again, say that we're looking for all files. We'll select the whale image, and since you would regularly have multiple images of the whale here, you could go ahead and select all, for example, 5 of the stills you extracted from this whale here. Once again, we'll be asked to enter information about the sighting. This is from our survey information, so sighting 1, and again, we'll just go ahead and say that this was from flight 1. And here we'll have to measure, or we'll have to enter again the same date as we have for the calibration flight, or the calibration information. Here we can enter identification information for the whale, and this is really important when you have a flight over more than one whale. So especially if you have ID information for these whales, or for example, if it's in a photo identification catalog, we can enter the identification information associated with that whale, and we can do this for each whale in the flight. Here we'll again be asked to enter that initialization height for the drone, so the difference between the takeoff height of the drone and the actual water level. And now we'll be asked to enter the altitude from that barometer reading. So that's again from the image, from the still image that we took. And so if we take a look at our whale still image, we can see that that barometer reading is 33.1 meters, which we can go ahead and enter here. Now we're going to be asked to bound the whale with a square and again crop the image so we can zoom in a bit here. So we'll right click and crop the image. Now we're going to be prompted to click one point on the nose and one point on the tail to create a horizontal axis for measurement. So this is not yet making measurements, this is just creating our horizontal axis that we're going to measure along. Next, we'll be asked to click one point on the left side and one point on the right side of the fluke. So our left side and our right side. Here we're going to again be asked to click 1 point on the nose and 1 point on the tail, and this is going to actually be our measurement of whale length. And we measure to the notch of the flute here. Now we'll be asked to, along the most visible side of the whale, outline the whale by clicking where each of these vertical lines intersect with the whale. So here we want to carefully click along the edge of the whale, the side of the whale, where it intersects with each of these lines. And it's important here to be as careful and precise as we can. Next, we'll be prompted to do the same thing on the opposite side of the whale. So now we're going to be asked to click 1 point on each side of the whale at where we think, as an observer, the widest part of the whale is. I'm going to go ahead and say I think it's right about here on this blue whale. And now what we're given is this measured image, and we're simply prompted to press enter twice to continue. So that completes our whale measurement, and just like with the calibration object, what gets sent to this exact same folder where the source of the image is a measured image and this CSV file. Here we can take a look at this measured image and a CSV file with all of the output information. So now to bring that information together, we go to R. So here, we're going to open up this file called whale quant analysis, and I'll go ahead and just show you this in the program file. So this is the folder that has all of the program information in it. We just use these MATLAB files, the whale calibration object and whale measurement. Now we're going to use whale quantitative analysis. That's an R file, and what's really important here is that this whale quant functions file is in the same folder as the quantitative analysis file. We don't actually need to open that functions script, but it needs to be in the same location as the analysis file. So now we can go ahead and go to this analysis file. First, it's just going to go ahead and kind of set your environment, set your working directory. We can go ahead and run that bit. Here you have the option to set what correction method you're going to use for the calibration, and for details on this you can refer to our publication on this method. That's Burnett et al. 2018 in Marine Mammal Science. And for this example I'm just going to go ahead and use method 1, but you can select which method you would like to be used here. There are some filtering options which I'm not going to use in this case. Here, if you have in GUI selected to set to yes, it's going to allow you to manually set the input directory, which I'm going to go ahead and do here. But if this gives you trouble, or if you're a Mac user, you can set your file directory, your file path right here to set your working directory. So since I have it set to yes, it opens up this directory kind of folder, and I'm going to go ahead and what we need to do here is select the file that's going to have all of those, the folder that's going to have all of those CSV files in it. It doesn't matter if they're in different subfolders, but you just need to select a folder that has all the calibration and whale measurement CSV files in it that you're interested in. Next, there's a series of packages that this is going to want you to load, and there's a version check. And if you haven't run this before, loading all of and installing all of those packages might take a little bit of time, just be warned. Next, all we need to do here is simply run this part of the program. If you have a lot of images you're running, this could take several minutes to run this part right here. And once you're done, what should happen is in that folder that you selected as your directory, you're going to have the outputs from this. And the most important one here is this morph measures folder, because this is what will have pulled together the information from the calibration scaling object with the whale measurement data. So this will give you the flight date, and the flight information, the whale that's measured, and all of the associated information, including surface area, which are these SAs, the percent of the length to width ratio at 20%, at 30%, at 40%, 50%, 60% along the whale's body length, the surface area of the whale, the body length, the body area index, and the whale length as well. That's that WL, and fluke width is FW. You can see that there are some NAs here because those are the lower and upper limits, and so if you measure more than one image for each whale but tag them all with the same whale identification information, this will generate confidence intervals and lower and upper limits for each of these measurements. And with that, that is the end of this photogrammetry tutorial. Again, you can find more information through the Geospatial Ecology of Marine Megafauna Lab's website. Thank you very much.